What are the different parts of the eukaryotic cell? How do all the parts work together? Where did they come from? When is the eukaryotic cell thought to have originated? Why do different parts of the eukaryotic cell have a distinctly different makeup to other parts? Over the next 3 explainer episodes, Amino Man and Gluco Girl are here to help you find out some more about the answers to these questions.
Welcome to this explainer covering eukaryotic cells, which in the AQA course is topic number 2. All of the notes and extra study resources can be found on Van Dyke Upper School Show My Homework. Choose the teacher filter of Mr. Chantrell. The first look learning video is also found on the Van Dyke Upper School Biology Vimeo channel. This video is the first of a 3-part series concerning structures found within the eukaryotic cell. This video covers the introduction to eukaryotes and is mostly concerned with membranes. Part 2 will cover the core parts of the cell. The more famous of the organelles which you are probably familiar with from GCSE, such as the nucleus. The concluding part of this series will look at the newcomers at A level. Those miscellaneous parts which you have probably never heard of up to this point.
All living things are made of cells, and cells are the smallest units of them that can be considered to be alive. There are thousands of different kinds of cell, but the biggest division is between the groups of cells that are described as being prokaryotic, the bacteria and archaea, and those being eukaryotic, covering such widely different organisms as animals, plants, fungi and protists. These are classed together as eukaryotic cells because of their shared common features. Prokaryotic cells are smaller and simpler than eukaryotic cells. They do not have a nucleus, and share fewer common features than eukaryotic cell types do with each other.
These key terms contain clues that can help you remember the 2 types. The use of the Greek term karyon denotes a center, or nucleus. Therefore, prokaryote means without a nucleus. Pro is a prefix used to mean before in biology. And eukaryote means with a nucleus. U is the prefix used to mean true. We'll examine prokaryotic cells in a different explainer, but in this video we will talk about the structure and function of eukaryotic cells.
Only in the past 70 years has an understanding of the eukaryotic cell emerged and the functions of its parts worked out. This has only been possible since the invention of the electron microscope, covered in the earlier explainer about the cell theory. This is the only imaging device with the resolving power to show up those parts lurking inside the cell, the organelles. Organelles, literally meaning many organs, do the jobs that make the eukaryotic cell come to life. From cell movement, made possible in the main by the cell membrane and cytoplasm, to respiration, taking place in the mysterious mitochondria. These tiny structures are literally the building blocks of cells, in the same way that cells are the parts that make up whole organisms.
Break it down even further, and the organelles are made of smaller and smaller parts too. Topic number 1 in the AQA course covers biological molecules, especially those that are known as macromolecules, and it's these tiny structures that when organelles are picked apart, we find that they are made from. Each organelle is a tiny specialized compartment in its own right, its particular specialization dependent on the biological molecules it contains. It is handy to think of the whole eukaryotic cell as like a carrier bag filled with jelly, and dotted throughout that larger bag are smaller bags that function together, making up the whole. This is what the organelles are like, literally bags within bags, each carrying out a separate, unique function based on its makeup.
The bag lining of these smaller and larger bags is where we start this whistle-stop tour of the organelles, which, as I am sure you know from GCSE, is known as membrane. Membrane is a true marvel of self-assembling design, partitioning up the cell up like a series of interior walls, and keeping the different organelles from interacting with each other, therefore increasing their overall efficiency. You will encounter membrane all over the eukaryotic cell, in virtually every organelle we study, often with strange and misleading names such as envelopes, cristae, thylakoid, lysosomes, etc., etc. But never lose sight that really this stuff is all the same, just named differently to confuse people like your good selves. The reason why membrane is so important is that is this organelle rather than the presence of the nucleus itself, that is truly the great separator between the eukaryotes and the prokaryotes.
We describe eukaryotes as having membrane-bound organelles. Even though prokaryotes have organelles, they lack membranes around them, hence why they are seen as a very different class of cell.
This will be added to in more detail in further videos, but as an initial introduction, membrane is constructed from a recipe of different proteins and glycoproteins. These molecules are combinations of short polysaccharide sugars joined to proteins. Also lipids and glycolipids, like glycoproteins, are combinations of short polysaccharide sugars joined to lipids. There is also a specialized lipid called cholesterol. And lastly, the key ingredient, phospholipids. All of these structures self-assemble into a 2-macromolecule-wide structure known as the fluid mosaic model. Although never seen in action by any camera microscope, this theory, put forward in the 1970s by Singer and Michelson, explains all the observed features that we know about membranes. The phospholipid ocean forms the bulk of the structure. The other parts mentioned can be added or taken away like boats in that phospholipid ocean, to make the membrane have the particular talents that the cell needs.
Depending upon the combination of the recipe that is put together, membranes can be fine-tuned to do a massive range of jobs. Want a membrane brilliant at exchange? No problem. Take out cholesterol and stick in more carrier and channel proteins. Less exchange and more rigid. Easy. Add cholesterol. Want more glucose from the surroundings, more than other cells can. Add more glucose carrier proteins. This is the principle that governs how the interior membranes of the cell are put together, such as the Christian nuclear envelopes.
Let's finish this explainer with a detailed look at the most famous of all the eukaryotic cells' membranes, the cell, or more accurately, plasma membrane. This layer forms the outside border of the cell, separating it out from the dangers that lie just beyond its borders. Everything inside it belongs to the cell. Everything outside is something else, namely the environment. It is incredibly thin, just 2 phospholipid molecules wide, with a range of other molecules extending away both inside the cell and outside into the environment at different points along its circumference.
Like any membrane, the plasma membrane is built according to the fluid mosaic model, and has a team of molecules within it. The exact mix depends on the cell type. They do tend to have a mix of parts that are generally suited to communication and exchange. More detail about these when we start to look at exchange and movement in AQA Topic number 3. Its roles go beyond this, and is an extensive list. Worth learning though. The importance of biological membranes is always a favourite of exam boards when setting essays for final exam papers. These roles are: number 1, selectively permeable barriers; number 2, structural, keeping the cell contents together; number 3, allow communication with other cells; number 4, allow recognition of other external substances; number 5, allow mobility in some cell types; number 6, the site of various chemical reactions.
Cells specialized for exchange or communication often tinker with their membranes to gain abilities, simply by adding in or taking away certain carrier and channel proteins, or adding in receptors to detect the presence of molecules for food or chemical messages, can totally change the job of a cell. This is the principle of how target cells work that respond to hormones like insulin, that you covered at GCSE. The cells of the liver and muscle have the correct receptors for that particular hormone, whilst the plasma membrane of other cell types lack the insulin receptor. Another example occurs in the cells of the small intestine. Cells in the lining often have many more nutrient channels and even active transport pumps to ensure that no valuable nutrients escape, increasing efficiency.
But where did this essential cell feature come from? Research over the past couple of years has suggested that the membrane is of primary importance in the evolution of the cell 4 billion years ago, and its amazing self-assembling abilities suggest that it perhaps came first of all the cell parts. Some data suggests that when the earliest cells were first evolving in hydrothermal vents, it proved crucial in not only enclosing the molecules of life, like nucleic acids and amino acids, but due to its special chemical abilities, it became a meeting point for all the other molecules of the early cell, herding them all together. Analysis of membranes of prokaryotes and eukaryotes suggests that there isn't only one type, and even more mysteriously, the eukaryotes seem to be more closely related to organisms that you know from GCSE as extremophiles, rather than the prokaryotes that often live on and inside your body, and sometimes even inside the cells themselves. Finished, at least for now. To continue this series about eukaryote structure, click on the link above for part 2.